Hello class, so we are going to talk about citric acid cycle, and this is a two-part series. So we're going to first start off with metabolism. And so as far as energy, the energy from the sun is traveling to the earth, and then with the plants, they perform photosynthesis, and then from there, we get a contribution of both carbohydrates and oxygen. And so that carbohydrates and oxygen gets broken down by heterotrophs. And from there, releases carbon dioxide and water, and also have a loss of heat, and thus feeds into the photosynthesis, and the energy once again is coming back from the sun. So it's this cycle that keeps on going over and over. And so this is where we're kind of talking about more so of this breakdown of the carbohydrates. So the citric acid is also called Krebs cycle, or it is called tricarboxylic, and is the hub of the metabolic system. It accounts for the majority of carbohydrates, fatty acids, and amino acid oxidation. And so the catabolism of proteins and fats and also carbohydrates happens in three stages of cellular respiration. And so the first stage is acetyl coenzyme is produced. The second step is acetyl coenzyme is oxidized. And then the third step is there is a electron transfer and oxidation phosphorylation. And so what you see right here is stage one, which is the oxidation of fatty acids, glucose, and some amino acids that ends up yielding acetyl coenzyme A. And so we already talked about with glucose goes through glycolysis to produce pyruvate, and then from there it goes through a enzyme called the pyruvate dehydrogenase complex, which ends up producing carbon dioxide and acetyl coenzyme A. Fatty acids, some amino acids also make the acetyl coenzyme A, but as far as the details for that one, that is more so for biochemistry part 2. And so on stage 2, when we have the acetyl coenzyme A, then it gets oxidized and thus goes through this citric acid cycle, and thus produce carbon dioxide and also NADH and FADH2, which are electron carriers that will be used for stage 3. And so with stage 3, these electron carriers goes to the respiratory system, and thus reduce oxygen to produce water. And also the electron flow also drives the production of ATP from ADP and having inorganic phosphate around. So as far as the citric acid cycle, it is found in the matrix of the mitochondria. So this is more so just speaking about the location of where this process is happening. So overall, the reaction catalyzed by the pyruvate dehydrogenase complex that has actually 3 enzymes within this complex, and what it does is take pyruvate, and it takes the carbon and the 2 oxygens that's highlighted, they get released to be carbon dioxide, and then the other two carbons gets attached to a coenzyme A, and thus making the acetyl coenzyme A. And so as far as cofactors for these enzymes is coenzyme A, which is really important. It's that thiol group. And then you have NAD plus, TPP, lipoate, and FAD. And this reaction also produces NADH that can also go into the respiratory and system and reduce oxygen to be water, and then facilitate that production of ATP. And so as far as this coenzyme A, this is what its structure is, but the main focus that we kind of look at is the reactivity of the thiol group, which has that attachment of the acetyl group. And then everything else is just more so this baseline definition of getting familiar with what is involved with coenzyme A and how it looks. So the process and mechanism behind producing, having the pyruvate and end up having that acetyl coenzyme A, the NADH, and also carbon dioxide to be released. What happens first is the first site, which is enzyme 1, which is the pyruvate dehydrogenase. So the fate of pyruvate is traced in red. In step 1, pyruvate reacts with the bound TPP, which is right here, and of the pyruvate dehydrogenase, which is that first enzyme, and is decarboxylated, which you see that release of carbon dioxide, to the hydroxyethyl derivative. So the rest of that carbon chain is gets attached to the TPP. So this first step is the slowest and therefore limits the rate of the overall reaction. Okay, then after we get done with step 1, now we're going into step 2, which is the transfer of 2 electrons and the acetyl group from TPP to the oxidized form of the lipolysyl group of the core enzyme. So this enzyme is dihydrolipol transacetylase to form the acetyl thioester of the reduced lipol group. And you'll see that happening right here, where you see that you have this dithiol bridge that ends up being reduced, and now you have that acetyl group is attached. And then from there, the residual amino acid for this is lysine. And so just having a little bit more information about this. Here you see lysine, and then from there we have this lipoic acid that has this bridge of 2 sulfur atoms, and then this is the form of it when it's reduced. The rest of the chain is just like this one. It's just conserving space, but just know that the rest of the chain is exactly like what is presented here. And then from there, that reduced form has attachment of the acetyl group. And so continue on with now focusing on step 3, is a transesterification in which the thiol group of the coenzyme A replaces the thiol group of the actual enzyme 2 to yield acetyl coenzyme, which you see right here, and the fully reduced right here dithiol form of the lipoyl group. And then from there, as we continue on, we got step 4, where the enzyme 3 comes into play, which is the dihydrolipoyl dehydrogenase, promotes transfer of 2 hydrogen atoms from the reduced lipoyl groups of the second enzyme to the F.A.D. prosthetic group of enzyme 3, restoring oxidized form of the liposyl group of E2. And you see that right here. You have that FAD. It gets reduced, and then therefore now we have the oxidized form of the lipolysin. And then from there, we have this FADH2, but that needs to be regenerated to FAD in order to keep the cycle to keep on going. And so that is step 5, the reduced FADH2 of enzyme 3 transfer a hydride ion to NAD+, which you see right here, forming NADH, which can enter the respiratory chain, and as once again reducing oxygen to water, and then promoting the electron flow of producing ATP from ADP and having inorganic phosphate. The enzyme complex is now ready for a catalytic cycle all over again. And so once again, you have that pyruvate. This group right here ends up releasing carbon dioxide, and then the acetyl group gets attached to TPP. That TPP gets attached. Now the acetyl group from there is the next step is reducing the lipolysin to have that acetyl group gets attached to this thiol group right here. That coenzyme A now replaces that thiol group and picks up the acetyl group, and now you have acetyl coenzyme A. You still have this reduced form. In order for it to cycle back to its oxidized form, it reacts with the third enzyme with FAD, which gets reduced, and in order to regenerate the FAD, the hydride is shared with the NAD+ that's outside here, which goes to NADH and goes out and be a electron carrier, and you have the FAD ready to have the cycle go over again. And this is just the structure of this pyruvate dehydrogenase complex, which is structured using a combination of methods, including X-ray crystallography and NMR spectroscopy. So you see you have this complex, you have E2 right here, and then you have E1 enzyme here, and the third enzyme over in this area, and then you have the lipoate, which is the lysine amino acids residue that has that dithiol group that is attached to it. And so this coenzyme A acts as a carrier of the acetyl group, which is a high energy, and thus eventually continue on through the citric acid cycle and produces other compounds. So this pyruvate dehydrogenase converts pyruvate to acetyl coenzyme A and carbon dioxide. And so what we're going to discuss in the next lecture is now that actual citric acid cycle. We just talked about the intro of what we need to do in order to get to the citric acid cycle. And so that's starting off with the pyruvate, which we talked about was produced from glycolysis. And what we discussed today in this lecture is how we got to that pyruvate to the acetyl coenzyme A, and now that feeds into the citric acid and start producing all of these NADH and carbon dioxide, which ends up producing also water and ATP. And this concludes part 1 of the citric acid lecture.